Welcome. Thank you all so much for coming today for this momentous event. And thank you to everyone on Zoom. I saw on Zoom there are people from India, there are people from Portugal, there are people from Germany. This is truly an international thesis defense. So we're all here today, as you all know, to watch Pallabi present all of the amazing research that she did during her time here at UNL. So I just want to give a very brief introduction, talk a little bit about her time here, and then I'm going to hopefully give her the full time to present her research to you. So for those of you who do not know, Pallabi is from Kolkata in India. Did I say that right? Okay. That's where she grew up. She went to the Heritage Institute of Technology, and she did some really interesting different research when she was there. She studied heavy metal contamination in wetlands and how they lead to resistant bacteria. She developed and characterized implant-grade bioactive glass. And then she developed, published, and presented a summer research project on ecological effects of spider mites. And this might be the one that is closest linked to her PhD research. But she came from India to UNL in 2017 and joined the School of Biological Sciences. But it wasn't until January of 2020 that she actually joined my laboratory. Now, if we think back to 2020 and the spring of 2020, this was exactly when everything shut down. So this Pallabi had just joined my lab. She had been working in the other section of our school, did not have a lot of background in ecology, evolution, certainly not in arachnids. And we had a whole project lined up for her that spring and summer, but then the pandemic hit and everything was shut down. We were not allowed to travel outside of the state. The university closed the doors and shut down. So in order to keep her research going, because this was really her first field season in my lab, we tried outsourcing some help from the public. We were looking for a particular species of wolf spider. We did not get any help from the public, but Dr. Rowan McGinley was still in the lab at that time, or was back visiting. And he is like the best person in the world. If you need to find a particular organism, ask Rowan and he will figure out how to do it. And it turns out that we actually have this species here in Nebraska, right near Branched Oak Lake. So Rowan helped us find a site for collecting. Pallabi did her first nighttime spider collecting with the help of other colleagues in the lab. We dedicated her to be the I forget what the title was, but like the one person from your lab that can come into the building during the pandemic. That was Pallabi, because she needed to run these experiments, but she had never done behavioral experiments before. She ran an entire set of experiments that summer, and in the end, there was some methodological challenges that led to none of those data being useful. So she spent her entire first summer getting a lot of experience in the field, having to figure all of this out on her own because she was the only one allowed in the building. But the next summer, we collected spiders for her in Mississippi. She took them to Cedar Point Biological Station, where she ran the experiment again successfully this time, and also started working on some of her other chapters. And the work that she did that summer is actually already published, her first chapter that she'll tell you about today. She also that summer worked on this incredible group of organisms that you will hear about today with Dr. Doug Gaffin, who's online right now, watching this presentation at Oklahoma University, and she's going to tell you about that work today too. But during all of this, there are several kind of qualities and traits that really became apparent about Pallabi. She is incredibly persistent. She is driven. She is resourceful. She's willing to try new things. She's ambitious, but she's also incredibly kind, generous, and creative. And I think there are very few people that could have switched entire research programs 2 years into their thesis, switched fields, switched organisms, switched everything, and still been able to do what she has done, which she'll be able to share with you today. During that time, despite the pandemic, we were still able to do some of our traditions. So she and Mike got quite good at carving pumpkins, making empanadas. She was around for multiple lab celebrations, some of which were masked, some unmasked, holiday parties. And she was even able to join the lab at the American Arachnological Society conference, where she actually won second place in the student oral paper competition, which was pretty phenomenal given that it was her first arachnology conference and her first time presenting research on spiders. It's also, I've been very fortunate that during the past 2 years when I've had this funding to run some of these 8-legged encounter events, Pallabi has been here. And it's been a time where everyone in my lab, and Brandy Williams, so a token Heavens Lab member, have really had opportunities to bond as we've traveled all over the state and country to deliver 8-legged encounter informal science fun activities to the public. So this is just a sampling of some of the honors and awards she has won during her time here. She received a research fellowship from the American Arachnological Society, from the Quantitative Life Science Initiative. This was during the COVID lockdown. She was a finalist in UNL Science Slam, vice president of BioGSA. She was the president of Toastmasters International, where she also has won some speaking awards, and the list goes on and on. She already has 2 publications, one of which is from her first chapter. Her second chapter is in review. And I just want to say thank you so much for joining the lab. You have added so much over the past several years, and we're really going to miss you. And with that, I will let you take over.
Thank you for such an amazing introduction, and thank you all for being here, both in person and on Zoom. Today, as you already know, I'm going to present my dissertation, which is titled Exploring the Function of Elongate Sensilla in a Wolf Spider and the Solifuge. Like humans, animals also use different types of sense organs and different types of signals to be able to receive signals from the world around them and also communicate with each other. These signals can be visual, acoustic, olfactory, or vibratory. And these signals can be used individually or in conjunction with other signals. So for example, male fowls use movements and calls to attract females. White-lipped ground-dwelling toads can inflate their vocal sac to create substrate bone vibration on the ground. And sea snakes can use visual and olfactory signals to find and hunt their preferred prey fish. Now, along with these signals, there are also some specialized sensory systems that some animals have that they can receive some specific information about their environment with. So for example, pit vipers can receive infrared signals from their prey using the pit organs. Dolphins have on their snout vibrisal crypts, which they can detect electromagnetic signals. And then Asian carnivores can produce a vibratory signal that is ultrasonic, so it cannot be heard by their predators, but it can be heard by their mates. And big brown bats can produce ultrasonic calls, and they can also receive those signals using their modified ears. Now, these kind of specialized sensory systems are also present in arachnids. For example, orb weavers use their webs sort of as an extension of their senses to receive airborne signals, airborne vibration signals. And leopards have modified long antenniform legs, which is their first pair of legs that's modified and has a lot of senses. And they can sense the world around them by taste, touch, smell, and they can use them for navigation or interactions. Scorpions on the underside of their body have these comb-like structures called pectins, and these are useful for receiving chemical signals and mechanical signals. And jumping spiders have really large anterior median eyes with which they can track and hunt their prey. Now, there are many different senses that might be very obvious to us, but there are senses which human beings don't have sense organs for. So they are elusive to us or hidden from us. And one of those senses is near field sound or air particle movement. So very close to the source of sound, the sound acoustic particle velocity and the sound pressure waves are not in phase. And some animals with certain sense organs can receive these signals as air particle movement. Humans receive sound as pressure waves, which is in the far field. So the near field is less than 1 wavelength long for any specific frequency of the sound source. And as I mentioned, these near field sounds can be received by things like trichobothria, which is present on different arachnids. These are long, thin sensory hairs that can receive this air particle movement signals and sort of move, which then travels, gives the information to the arachnid about their prey or predator or anything else around in their environment. Now, air particle movement signals have been studied in arachnids. For example, they have been studied in agonistic interactions in amblyphigids. Amblyphigids can actually generate these air particle movements by moving their antennniform legs really, really fast. And they can be received by their opponents, trichobothria. Now, it was seen that when you cut out the trichobothria of 2 interacting amblyphigids, their contest durations became longer, and it would often turn violent. And another example is for the tiger bromeliad spiders, which can use these signals to hunt their prey, which is over 25 centimeters away. They can feel the wing flaps of tiny insects, and their trichobothria can also provide them with information about which side, the direction of the signal, and also these trichobothria can respond to a broad range of frequencies. Now with that, I come to my first study organism, which is Schizocosa retorsa. And these are found all over US and Mexico. They are they live on dry areas, on grass or sand, and they do not build webs. They hunt their prey on the ground. And they have these long, thin hairs on their legs. They are all over their legs, and it is possible that they can receive air particle movement signals using these trichobothria. And we don't know a lot about how these signals might be used in courtship rituals of these spiders. So that is a very interesting point. My second animal is solifuge, or Eremobetes polypus. And these are found in tropical and temperate deserts all around the world. And they are covered in hair. They're very fast. They're very difficult to keep alive in the lab, but they have a lot of hairs or sensilla on their body. The ones that I'm specifically interested in are on their 4th pair of legs. Here you can see with the arrows here. They're called elongate sensilla. And previously it was thought that these elongate sensilla and the trichobothria are the same thing. But recently with morphological studies, it has been seen that they are not the same. So here's an example of a trichobothria. As you can see, the base of the trichobothria is cup-shaped, which makes them very sensitive to minute signals like air particle movement. On the other hand, the elongate sensilla has a socketed base, which means that they can receive mechanical signals, but we don't know how sensitive they are to these signals. Now, with all that in mind, I will give you an overview of the 3 chapters of my talk. And notice this leg of an arachnid, which has 3 parts. So each of the parts will sort of correspond to the 3 chapters. So my first chapter is uncovering hidden signals, previously presumed visual signals likely generate air particle movement. This was published in Frontiers in Ecology and Evolution. And whenever you see the first part of the leg sort of highlighted, I'm talking about the first chapter. My second chapter is potential role of sensilla in the reception of air particle movement during wolf spider courtship. And this chapter is in review with Journal of Arachnology. And then my third chapter is electrophysiological response of elongate solifuge sensilla to air particle movement and air pressure stimuli. And I also intend to publish this chapter as well. So you will see this leg on the bottom right corner throughout my presentation, and whichever part is highlighted, that is the chapter that I'm talking about from left to right. Let's get started with the first chapter, uncovering hidden signals. Previously presumed visual signals likely generate air particle movement. Now here we see a male Schizococeratosa, which had one of its front legs raised up, which then it will bring down in a wave. And they can do this really, really fast in 1 second, multiple times in 1 second. So we'll watch a video here. So what we saw in this video is the courtship behavior of these male Schizococeratosa wolf spiders. They do some push-ups, some purple drumming, and then some leg waving that consists of multiple individual leg waves, but they do it in parts, which we call bouts. And these bouts can be interrupted by push-ups or moving or anything else that they're doing, but not moving their legs. Now, in previous studies, it was seen that even though visual and vibratory signals are very important for related species of these spiders, it is not as important for these spiders as they can mate just as successfully in the presence or absence of visual or vibratory signals. And it was also seen that more leg waving from the males led to a higher mating success. And this leg waving, because it's so fast, it can be on more than 13 times per second. So it can generate air particle movement signals that will be received by the female's Trichobothria. With that in mind, we came to the question, what is the importance of near field sound in the mating success of Schizococeratosa wolf spiders? More specifically, will the disruption of air particle movement affect their mating success? Now, to answer this question, we set up this arena where we have an arena on top of a granite slab. Now, granite does not transmit their vibratory signals. And on top of that, we have a speaker. Now, this speaker can play sounds of different frequencies, but we settled on playing 100 hertz. Now, we have two different kinds of speakers. One speaker with the speaker cone removed. Now, when this speaker was playing, it would have all the different characteristics of the speaker being played, but it wouldn't produce any noise or disturbance in the near field of the spiders. But when we play the speaker with the speaker cone intact, it will produce noise or disturbance in the near field of the spider. Essentially, the sound coming off the speaker cone intact, that frequency will trigger the Trichobothria of the spiders, of the female spiders, but that would compete with the signals that the male are producing, and likely there will be some effect on their mating success due to that. Now here's a video of when I was playing 100 hertz through the speaker with the speaker cone intact, within one wavelength of the frequency, and you can see the Trichobothria is getting triggered and it's moving. So that means that our experiment design works. We also looked at the Trichobothria when the other speaker was playing and saw that there was no movement in the Trichobothria. And all these, the whole experiment was done in the dark because we didn't want the spiders to have any visual signals either. And the whole experiment was recorded using infrared camera. And I later analyzed those videos. So the data that I collected from these videos are first and foremost whether they mated or not, whether they successfully mated or not. The next is latency to mate. Now, this is defined by when the males actually started courting during the whole time, and then up to the time when they successfully mated or till the end of the video. Next, we recorded some courtship behavior that we saw in the video. So leg waving bouts, number of leg waving bouts, number of individual leg waves, and number of push-ups. And then we did some calculations where we found out the rate of each of them. So the rate of leg waving bout is equal to the number of bouts divided by the latency to mate. And similarly, we calculated the rate of individual leg waves and the rate of push-ups. Now, this leads to our results. where the first thing we saw is how mating success varies in the different signaling environments. So here on the x-axis, we have the two different signaling environments, and the y-axis, we have how much percentage mated, and each of the group had 26 pairs of male and female spiders. So what we saw using a chi-square test is that there was a significant difference between the pairs that mated when there was no noise or no disturbance in the near field versus when there was disturbance or noise in the near field. Now, next we wanted to check if there was a difference in how fast they mated. Did one of them mated faster than the other group? So we did a Kaplan-Meier survival analysis plot, and here the green is where there was disturbance and they took longer to mate, while the no noise or no disturbance is the blue one, where they took less time to mate. That means there must have been a lower signal to noise ratio in the noise environments, which prevented the spiders from mating faster, even if they mated. Next, we looked at the rate of courtship behavior, which also varied in the different signaling environments. So here we are looking at the two different signaling environments on the x-axis and the rate of leg waving bouts on the y-axis. And we also looked at similarly at the rate of individual leg waves and the rate of push-ups. So what we see here is that there is a significant difference between the leg waving bouts, rate of leg waving bouts in the no noise environment versus the noise environment, which means that the male, it was altering its behavior when there was noise or disturbance in the environment. And we see similar results for the other courtship behaviors, rate of individual leg waves, and the rate of push-ups. Now, we mainly focus on the rate of leg waving bouts and the rate of individual leg waves, because that is the action that can potentially generate the air particle movement signals. So next, we did a model with the binomial logistic regression, and we saw that there was no interaction between the signaling environments and the courtship behavior, but there was a significant effect of the courtship behavior on the mating success. So next we looked at the rate of courtship behavior, how they varied with mating success. So on the x-axis here, I have whether they mated or not, so yes and no. And then on the y-axis, we have the rate of leg waving bouts. So here we see that the males that mated had a higher rate of leg waving bouts than the males that did not. And this goes on to say that the rate of leg waving predicted mating. So the females were assessing the males on the rate of leg waving, and they chose males that had higher rates of leg waving. So to conclude, the first thing I found is that the presence of noise in the near field resulted in reduced mating success. Noise altered the male's courtship behavior. We have seen there has been papers about behavioral plasticity in males, in other animals, and it is possible that the males think of their courtship behavior as a costly, energetically costly behavior. So they don't do that behavior as often if there is an environmental noise or disturbance in the environment. And the rate of leg waving was significantly higher for males that mated successfully. So since the rate of leg waving predicted mating even without visual or vibratory signals, we can say that there is something else that the females are assessing the males on. And since the rate of leg waving did predict mating and the leg waving can produce air particle movement signals, we think that the air particle movement is the signals that the females are assessing the males on. So all that we learned in our first chapter leads to my second chapter. Here we want to find out how the females are receiving these air particle movement signals and if Trichobotria is important in this role. So my second chapter is the potential role of sensilla in the reception of air particle movement signals in wolf spider courtship. So right away we come to the question, with whatever we have learned from our first chapter, is that what is the role of female Trichobotria in the mating success of the Schizobosa retorsa of wolf spiders? Now, to answer this question, we wanted to modify or manipulate the Trichobotria on the females. So we had one group of females where the Trichobothria was intact. We did not do anything to the Trichobothria. And then we had a second group where the Trichobothria was trimmed or ablated on the first two pairs of legs. Now, to do that, we used a technique where we placed the female spider on the sponge and then put a cheesecloth on top of the sponge to trap the spider, and then use entomologic pins around the spider to keep it in its place. And then we carefully use the forceps to guide the first two pairs of legs out through the cheesecloth. And then we put the whole setup under a dissecting stereoscope and then use the microserrative scissors to cut the Trichobothria. So here's what a spider looked like when it was in this process. None of the pins are hurting the spider. It just needs to be trapped so that it doesn't hurt itself and I can cut its Trichobothria properly. So we ended up with two groups where for the unablated, they did go through this process, but their Trichobothria was left intact. They were held in this under the cheesecloth for a few minutes and then let go. But the other group, the Trichobothria ablated or trimmed group, we actually cut the Trichobothria after putting them in this position. And then later, when we counted the number of Trichobothria left on the first two pairs of legs of each of these spiders, we saw that there was a significant difference in the number of Trichobothria present, but we did not get rid of all of the Trichobothria in the trimmed group. And this experiment was set up similar to the first experiment, where we still had them on a granite platform so that they didn't have vibratory signals. It was the circular arena in the dark, so they didn't have visual signals. But this experiment did not have any of the speakers. It was just the females with the Trichobothria intact or the Trichobothria ablated. And we recorded this with the infrared camera and then collected similar data to the last experiment from the videos. So as a result, we looked at the mating success of these spiders in the different signaling environments. So here on the x-axis, we have the two groups, unablated, Trichobothria intact or ablated. And then on the y-axis, we have the percentage that mated. And our sample size was 15 pairs per. And as you can see, there was no significant difference between the two groups. They mated just as successfully in both the groups. We checked to see if any of the groups mated faster than the other groups. So we did a Kaplan-Meier survival analysis plot, and they overlap very well. So they did not mate faster or slower. Each of the group took similar amounts of time for their mating. So we looked, then we performed a binomial logistic regression model, which the model itself was not significant, but it suggested that there might be an interaction between the two different groups and their rate of leg waving bouts. So that's what we looked at. We have on the x-axis, the unablated and the ablated groups. On the y-axis, we have the rate of leg waving bouts. And in the unablated group, they had a significant difference between the rates of leg waving bouts for the ones that mated in orange and the ones that did not mate. So this goes along with what we found in our first experiment, that if they wave their legs faster, they will have higher success in mating. But in the ablated group, there was no significant difference between the rate of leg waving. There is a slight trend, but there was no significant difference, which means that the females could assess the males using their Trichobothria, because their Trichobothria was intact, based on the leg waving rates, but they could not do the same assessment when their Trichobothria were cut off. Now, to conclude, despite there being no significant difference in the mating success, the leg waving rate can only predict mating when the Trichobothria are intact. And thus, air particle movement is an important mating signal that can be assessed by the females to make mate choice decisions. Now I have future directions for the first two chapters together. So one of the things we can do is use electrophysiology to understand the female sensory reception of air particle movement, to see what the females are actually detecting when there is air particle movement signals. We should also check the role of male Trichobothria, since the males did vary their courtship behavior when there was noise in the near field in our first chapter. So the disturbance affects the males in some way, maybe because of environmental noise, or maybe they think there's other males nearby. They have some sort of effect, so we can also check the role of male Trichobothria in receiving air particle movement or near field sound. And for the third chapter, because second chapter, since they have a lot of Trichobothria on their third and fourth pair of legs as well, we could try different techniques to maybe able to ablate or deactivate all of the Trichobothria on the females. But it would be an intricate process because we don't want to accidentally snip off any part of the legs or force them to let go of their legs or autotomize their legs. So that would be something that would be interesting to look at. With that, I change animals. We come to our third chapter where we're studying solifuges, which is titled Electrophysiological Response of Elongate Solifuge Sensilla to Air Particle Movement and Air Pressure Stimuli. Now, to reacquaint you to the animal we are talking about, these are very fast-moving, desert-dwelling solifuges, which are very difficult to keep alive in the lab, and that dictated what kind of experiments we did with them. So specifically what we are studying is their elongate sensory hairs on their femur and tibia. So we started with this broad question because these species is very understudied, so we don't know a lot about a lot of their sensilla. So what is the function of this elongate sensilla on the fourth pair of legs on solifuges? And to answer this question, we used a process called electrophysiology, which can get the electrical signature of the cells and see the cells that are connected to the hairs respond to different kinds of stimuli. So here we have the whole setup inside a Faraday cage that gets rid of electrostatic and other interference from electrical sources. We have them on a vibration dampening table, so in case while working we bumped on the table, the electrode doesn't get messed up. And here we have the solifuge on a slide on a micromanipulator. Now here you can see that they're put together, the solifuge is put in its place using modeling clay, but this was our first trial, but by the end of it, we used up something like this. So this is a view from the bottom, and this is a view from the top. We used a lot of modeling clay to keep the solifuge in its place, and we also used some double-sided tape so that the solifuge was not moving. But even after all this, the solifuge was trying to chew its way out through the modeling clay. That is on a micromanipulator under a dissecting scope so that we can focus on the specific sensilla that we're looking at. And this other micromanipulator has an electrode. So the micromanipulатors help make small movements of the slide and the electrode. And we look through the dissecting stereoscope and make sure that the slide is actually at the base of the elongate sensilla. The electrode is at the base of the elongate sensilla on the solifuge. And then we also use an indifferent electrode, which is connected to a different leg on the solifuge so that it completes the circuit. Now, all of this is recorded using an amplifier, which connects to an oscilloscope, which connects to an analog to digital converter, which connects to a computer that has the software Spike 2 that I used for this process. So anything happening here is getting recorded in that software. Now, if you want a closer look, so this is what the first solifuge looked like when we put the electrode at the base of one of its hairs. So this is a closer look through the scope. So we have the electrode at the base of the long sensory hair. Now, since initially there were some similarities that scientists thought that the Trichobothria and the elongate sensory hair are sort of similar, and because these hairs are also long and thin, we decided to test the air particle movement stimuli. And for that, we used the speaker cone that had the speaker, that had the speaker cone intact, and that was also placed very close to the solifuge, about 10 centimeters away, so that all the frequencies we played through it had a near field effect on the hair. And then we also used an air pressure stimuli to check its sensitivity. Now, the first, we checked the air particle movement stimuli. We used frequencies from 10 to 1000 hertz. For a few of the animals, we went from 10 to 300 hertz at 10 hertz increments. And then beyond 300, we did 50 hertz increments. For others, we just did 10, 50, and then 50 hertz increments right away. And then all of these, the speaker was placed 10 centimeters away to make sure there was a near field effect. So here is a reminder of what these long sensory hairs, here Trichobothria, looks like when it's getting triggered by near field sound or air particle movement. So as you can see, when we play these signals, they are moving and they get triggered. But when we placed these solifuges under a similar effect, try to look at these long sensory hairs and see what they do. As you can see, these hairs are not moving at all because these hairs are likely not responding to air particle movement, but we cannot make these assessments just by visually looking at it. That's why we did electrophysiology. Now I have some example electrophysiology data. So when electrophysiology gets recorded, it looks something like this, where each of these spikes are where a stimulus was applied, and so it increased a spike beyond the baseline, and then the response went down as the stimulus went down. So this is what a raw recording would look like. And then when we sort out the spikes from this, it would look something like this. So these spikes don't have the background spikes, it's just the spikes that are in the response. And from these electrophysiology data, we can also quantify the average number of spikes per second from a data like this. And we can also find out the number of spikes per second, which I will explain in a little bit. So the electrophysiological responses to the air pressure stimuli, air particle movement stimuli. Now, first, I'm showing you the control. The control is to a location where nothing was playing. So we took a part of the recording where nothing was playing, and that is the control. So we had several controls. I chose one that had all the different spikes that we saw throughout the recording. So here are 4 different kinds of spikes, and then here is the waveform of the spikes. So when we superimpose these spikes, they look like this. So we have a blue one that goes like that, a sky blue one, green one, and a purple one. And these recordings also had some taller spikes. You can see the difference in the voltage here. And these taller spikes, there were 2 of them, and this is their superimposed waveform. Now, next, I will show you representative data of 10, 50, 100, 500, and 1000 hertz. And what you will see is that there's just maybe a subset of all these spikes on all of them. There's no new spikes, and there's no increase in the spike, average number of spikes per second or anything. We cannot see any difference in the spike patterns from the control with the different frequencies. So we quantified that with the average number of spikes per second. For that, we took a 30-second portion, and we got the average number of spikes per second over that 30 seconds. So on the x-axis, we have here the control and the different frequencies, and the y-axis, we have the number of spikes per second. And by using an independent 2-sample t-test, we saw that there was no significant difference between the control and the frequency groups. We also applied a linear regression line on the frequency results, and there was no significant slope of this linear regression. So from this, we realized that these hairs are not responding to air particle movement stimuli. So next, what we tested is air pressure stimuli. And for that, we used forceful breath, essentially blew on the hairs. And this, initially, we did some of the experiments where we were just looking for response. And then we did some experiments where we looked at, we applied the stimulus at 3-second intervals. So we'll apply the stimulus, wait 3 seconds, and apply it again multiple times. Same for 2 seconds, apply it, wait 2 seconds, then apply it again. And similarly for 1 second. We also applied a prolonged stimulus where we would apply the stimulus for 3 to 4 seconds at a time and then look at the response. And all this, the forceful breath was applied from 23 centimeters away. So again, we will look at a video where these are the sensory hairs, and I'm essentially blowing at these hairs. So look at the hair, what the hairs are doing. You can see that they're moving right away. So it is possible that these hairs are responding to the forceful breath stimuli. So now we will look at the electrophysiological responses to the air pressure stimuli. So here we have the raw recordings. We have the responses at 3-second intervals, 2-second intervals, 1-second intervals, and the prolonged. And in between these stimuli, these are the control regions that we used as control because there was no stimuli applied to them. And when we sorted the spikes from this, we found some smaller spikes and some taller spikes. So by superimposing the smaller spikes, this is what they look like. And by superimposing the taller spikes, this is what they look like. And we were curious whether these are, what is the origin of these? Are they coming from separate sources or the same sources? So we performed autocorrelations within the smaller spikes, and we found out that all the smaller spikes are from the same cell. And then we did autocorrelation within the taller spikes, and also we found that all the taller spikes are from the same cell. And then when we cross-correlated the smaller and the taller spikes, we found that the smaller and the taller spikes are from the same cell as well, which makes sense because the smaller spikes start appearing when the taller spikes get smaller. Now we quantified this by taking 1-second bins from when the stimulus was applied and finding out the number of spikes per second from that. So here there are like 10 instances of the 3-second interval, 10 or 12 of the 2-second intervals, and so on. So we averaged them and looked at the results. So here on the x-axis, we have the control, 3-second, 2-second, and 1-second intervals. And on the y-axis, the number of spikes per second. And unsurprisingly, they were all significantly different from the control, which makes sense because there was actual response to these stimuli. But surprisingly, we saw that there was a significant difference between the responses at 3-second interval and the 2-second interval, which means that the response is getting adjusted or sensory adaptation is occurring when we are applying the stimulus at 3-second intervals, which gives them time to recover, which gives the cells time to recover, versus at 1-second interval, which doesn't give the cell a lot of time to recover. So we think that the recovery period of these cells are anywhere between 3 and 1 second. The cells need some time to be able to recover to respond at the same rates again. And next we looked at the prolonged stimuli. So here on the, we collected data for 1 second before the stimulus was applied and 4 seconds after the stimulus was applied. And remember, the continuous stimulus was for about 3 to 4 seconds. And then we used a rolling average method to see any trends for the spikes. And this is what we see for the 3. So as soon as the stimulus is applied, there's a spike in response, and then the response slowly goes down, which means that these cells are adapting to losing the ions, trying to respond to the stimulus. And when they'll have lost all the ions, they stop responding. They need time to be able to recover and respond to these to the stimuli again. And then we performed a passive breath stimuli, which was like this, and that is to control for the forceful breath stimuli. It would control for any temperature changes that the forceful breath stimuli was introducing, any humidity changes, or anything that the solifuge might be reacting to if I ate something during lunch that they didn't like. So this was a control for the air pressure stimuli. And what we see is that these are the arrows when the stimulus was applied, and we see no spikes or no response to the passive breath stimuli. So the response that we see to the air pressure stimuli is due to the forceful nature of the breath and not temperature, humidity, or chemical signals. Now, to conclude, elongate sensilla on the fourth pair of walking legs are not sensitive to air particle movement stimuli, but they are sensitive to air pressure stimuli. Now, we saw that these responses are fast adapting and fast recovering. And by adapting is just the adjustment of the response, sensory adaptation. And then we also saw that the response was all from a singular cell at the base of the hair. And then this was also a phasic mechanosensory response, which means the response peaks right when the stimulus is applied. And then with continuous exposure to the stimulus, the response goes down. And then it needs some time to recover to be able to respond again. And we think that the response, the solifuges can respond to the air pressure stimuli because it's a predator escape response. It has been, solifuges live in burrows in different desert areas, and they are known to be predated on by different kinds of birds. So anecdotally, they have been seen to be hanging out outside their burrows with their back legs sticking out, and the hairs will come out this way. So it is possible that they are able to detect wing flaps of a bird, the air pressure that produces, and be able to escape that predator. Now, for future directions, behavioral trials would be very important to understand how exactly the solifuges respond to the air pressure stimuli. We can also test, because the solifuge is covered with sensilla, we can also test its other hairs, maybe some other hairs are responding to air particle movement stimuli. And we did not investigate whether these hairs were also responding to temperature, humidity, or chemical signals, except for what I introduced. So maybe they're responding to, they can respond to something very specific, and we haven't tested for that. With that, I would like to provide a summary of my whole dissertation. So from the first chapter, we learned that noise in the near field disrupts mating success in the Schizocosa retorsa spiders, along with altering the male's courtship behavior. From the second chapter, we learned that the rate of leg waving influences mating success when females have intact trichopotria to assess the male's courtship behavior. And then from the third chapter, we learned that the elongate sensilla on the solifuges can receive mechanosensory air pressure stimuli, but they are not sensitive enough for air particle movement stimuli. And to conclude, there are modes of communication in arachnids that are beyond human perception, yet they might be very important in understanding how they're using their different modes of communication together to transfer information or receive information. And then understudied sensory structures can provide us with valuable information about these arachnids, their evolution, and how their communication evolved. And last but not the least, arachnids are super cool. With that, I would like to thank everyone who has been helping me throughout my journey of my graduate career, Dr. Hebetz, definitely, and then my committee members, John DeLong, Dai Shizuka, Ian, and Troy, and then Dr. Doug Gaffin at University of Oklahoma Norman, who helped, this whole electrophysiology was done at his lab, and he helped me out with analyzing the data. That was a lot of work, and I really am thankful to him. Hebetz lab members who not only helped with moral support, but also feedback for different manuscripts, and visitors at Hebetz lab, Franco, Guy, Mariella. And then Ryan and Erica are very important because they are the ones who collected the solifuges at the Rocky Mountain Arsenal Wildlife Refuge and overnighted them to Doug's lab to be able to do this experiment. And also UNL Behavior Group for all the snacks and feedback. And these are some of my funding sources. But along with everyone who has been involved in my academics part of the graduate career, there have been many people who have helped me out with just staying sane throughout this process. So my family, my nephew, this was the last time I saw him, he's double the age now, if you'll believe it. And my niece, who I have not seen in person yet. My friends, my cousins, my friends from school, Toastmaster, Mike, and Amy, my sister and my parents here. And then these are some of the pictures from the lab that you saw. I have one of the pictures with Doug as well when we visited, when I went to his lab. And these are just very good memories that I made in my graduate career at UNL. With that, I would like to take any questions.
I'd be curious what's known about variation in the number of these structures, either like between the sexes or between different species, or even within species. Did you notice that individuals vary in the number
Of the elongate sensilla?
or the Trichobothria and that?
The Trichobothria we did calculate, and there was anywhere between, for the first two pairs of legs, there were anywhere between 22 to 40 Trichobothria per leg.
So they vary.
So they vary quite a bit, yes. And for the solifuges, no, we only had a few animals to do the electrophysiology from. They each had 2 to 3 elongate sensory hair in each part of the leg. So the tibia had 2 to 3, the femur had 2 to 3, and so on. But I did not compare between sexes for that.
But they're not known to be sexually dimorphic?
Not these hairs that I know of. It's very understudied. There's not a lot of stuff done.
Yeah, I was just curious to know, because you're sort of associating them with response to male signaling in the
For
wolf
the-
spiders. So I'm just curious, like, do males also have them?
Males also have Trichobothria, but I have not quantified them. There is a possibility, of course, there might be some variation, but I don't know exactly.
The amazing experiments where you were giving spiders haircuts. Am I right that you only shaved the females,
Yes.
but you saw that the males changed their courtship behavior? What's up with that?
Yes. So what we think is that because there was noise in the near field, maybe the males are responding. Males are not doing an activity that is energetically costly to them, when they think that the females might not even be able to receive that signal. So that's why they're doing it less, and that's why there's less mating success. But both of these experiments were done around at the same time. So I did not follow up on the male side of this equation, and that would be something very interesting to be able to look at.
So I thought that the males behaved differently, not because of the sound, but because they were presented with females that had or had not had their Trichobothria cut.
You're
confusing
combining
the
two different
Okay.
chapters.
It was just the sound where they
The
changed
sound,
behavior.
the noise,
Okay,
yeah.
that makes more sense.
I kind of have like a follow-up question on that. At the end of your chapter 2, that plot where you have the interaction. So in that experiment, there was no sound playing. when
No,
the
no
females.
sampling.
But then the females who had the Trichobothria cut, the males that were paired with those females did show different behaviors, right?
No, there was no significant difference in the rate
Oh,
of
and the
liquid.
non-ablated ones. So the ones that have pairs, some males courted more than others.
So the
So
males
basically
that
they're also
mated
used by that.
successfully courted more, which means that if the males were courting more, there was a higher chance they would be able to mate. But we didn't see that difference when the female's Trichobothria was cut off. So I think here what we can say is the female assessment. So the female are choosing the males based on their rate of leg waving when their Trichobothria is intact, but they are not being able to do that kind of assessment when their Trichobothria is cut off.
So when the Trichobothria is cut off, there was not a significant difference in the amount of leg waving between males.
There was no difference between the males that mated versus that did not.
So
That would probably be a good pair, like just think about your graphs, because I think that was the confusion for both of those questions. So just having something that said that the treatment of females doesn't affect the leg waving rate.
Okay.
And then Doug has a question.
In this case, I don't think there will be a difference because the recording of the male will not be able to produce any air particle movement that the female will be able to sense with its Trichobothria. Is that what you're asking, Doug?
Yeah, well, I guess I was asking if you could deliver a similar stimulus, a vibratory signal somehow without the male being there. I'm not sure how you do that, but
We can do that with the speakers, do the exact frequency that the males produce and see if it.
Right.
Do you think the solifuge cannot receive near-field sound, or is it possible they use different types of ears? This is very interesting. Yes. So what I think is that the solifuge cannot receive a near-field sound using the hairs that I tested, but it is possible because, like how hairy they are, they have a lot of hair, and it is possible that there is some other hair that might be doing that more. So yeah. Thanks, Nori.
Anyone else online, in the room?
I have a silly question. How long, if they did, so do the trichobacteria, do they grow back after you cut them off?
Not for adults.
Not for, oh, so that just, they're just short. Forever.
Yeah.
Okay.
But these spiders only have a 1-year lifetime.
Okay.
So after doing the experiments, I preserved them, like froze and preserved in ethanol anyway. So yeah.
Ian has a question.
Do you think the spiders with less reliance on passive webs would tend to possess more hair, or hairs replacing webs in evolution? That's very interesting. There have been evidence of some spiders using their webs to sense airborne vibration, and with spiders that do not have webs, like bull spiders, it is possible they're just using their hairs to receive these airborne signals like air particle movements. And that would be an interesting question to look at. Are webs replaced by hairs in these spiders? Thank you.
Any other questions? Okay, thank you all for coming. Congratulations.